co-chair is presenting remotely, so I'd like to introduce him. That's Antoine Garnier-Coussard. He is a geriatrician at the Clinical and Research Memory Center of Lyon and a PhD candidate in the Châtelet Lab in Lyon. And so hopefully we can load him up. I'm supposed to tell everyone who's interested in CMEs that there's a sign-up sheet on your way out if you'd like to sign up for CMEs. So could we load up Antoine? There we
Hi,
go.
good afternoon, everyone. Can you hear me?
Yes.
Okay, that's great. I'm more than happy to be here and to chair this session with Adam Brickman, who is professor of neuropsychology at Columbia University. And we're going to talk about white matter hypertensities in Alzheimer's disease. I'm really sorry I can't be with you in person. I'm in France currently, but I promise I'm not at the Olympics. White matter hypertensities, as you know, are typically considered as vascular lesions, leading to the vascular contribution of cognitive impairment and dementia. These vascular lesions arise from hypertensive angiopathy as well as cerebral amyloid angiopathy, or CAA. However, emerging evidence emphasizes the heterogeneity of white matter hypertensities pathophysiology, suggesting non-vascular mechanisms, particularly in Alzheimer's disease. This has led to the alternative hypothesis that some white matter lesions may be secondary to non-vascular AD-related processes. We think it's crucial to unravel the origin of white matter hypertensities to improve individual diagnosis as well as precision medicine. Moreover, as CAA increases the risk of adverse events from anti-amyloid drugs, notably amyloid-related imaging abnormalities or ARIA, and as white matter lesion is now an official feature of CAA according to the more recent Boston criteria, we can hypothesize that in some cases, white matter hypertensities may be a risk factor for ARIA. In this perspective session, we will aim to provide an overview of the pathophysiology of white matter lesions in AD, covering both vascular and non-vascular pathways, and we will also aim to offer guidelines for the clinical interpretation of white matter lesions in AD patients, and especially at the era of anti-amyloid drugs. Firstly, Adam Brickman and I will discuss evidence supporting the vascular and non-vascular origins of white matter lesions in AD. Then Masadada, who is present in person with you, and she is assistant professor at Douglas Research Center, McGill University, will discuss the contributions of neuroimaging tools to better understand in vivo the origin of white matter lesions. Finally, Andreas Charidimiu, who is also virtually connected, he is assistant professor of neurology at Boston University and neurologist at VA Boston and a specialist of CAA. He will discuss the relationships between white matter lesions and CAA, as well as the risk of ARIA in the case of CAA with ischemic white matter features. And finally, he will provide a perspective on the benefits and risk of anti-amyloid drugs in the case of white matter lesions in AD. I hope you will enjoy this session and you will find it interesting. And we encourage you to submit questions through the app as well as the virtual platform. And don't hesitate to ask any questions you may have, especially at the end of the session when there will be a Q&A session. So now I'm going to introduce again the first speaker, Adam Brickman. I thank him very much to accept to participate in this session and to co-chair this session. He's a great and specialist of white matter lesions and of Alzheimer's disease and a professor of neuropsychology at Columbia University. So I'll let him start the first talk, and I wish you a very great session.
Thanks so much, Antoine, for the introduction. I showed one of my students my slides, and she asked me if it was a full-day workshop after I went through all 120 slides. So I tried to cut back, but hopefully we only have 4 speakers, so I'm going to be a little bit casual, I think. So I'll be talking about the association between white matter hyperintensities and Alzheimer's disease, and whether this, in fact, suggests a pathogenic role of vascular disease in Alzheimer's disease. So these are my disclosures. I'd like to just point out that we formalized some of these thoughts in a piece that I wrote with Batul Rizvi that was published in Alzheimer's and Dementia this year. So if you want to see that. So what are white matter hyperintensities? I think if you're here, you probably know about them. They're these increased signals on T2 weighted MRI scans, such as these fluid attenuated inversion recovery or FLAIR images you see here. Lots of labs have methods to quantitate these lesions and make 3D animations to impress their audiences. White matter hyperintensities typically are at least in the context of aging, are of presumed vascular origin. They can regress, they can stay stable, they can expand, they could lead to incident infarcts, or they could cavitate. And typically, at least clinical radiologists often attribute these lesions to arteriosclerosis, but there are also many other vascular components that give rise to white matter hyperintensities, including dysfunctional endothelial cells, dysfunctional oligodendrocyte maturation or oligoturnover, and astrocytic abnormalities. So we and other groups, notably Charlie DeClarley before us and others, have done a lot of work connecting white matter hyperintensity, volume or severity to late onset Alzheimer's disease. And we typically see something like this: people at risk for Alzheimer's disease by virtue of having amnestic MCI have increased white matter hyperintensity volume. If we look regionally by lobe, we know that people with prevalent clinical Alzheimer's disease or amnestic MCI have a propensity for higher white matter hyperintensity burden in posterior areas, predominantly the parietal lobes, but frontal lobe white matter hyperintensities seem to go with general cognitive decline and aging. Charlie's group publishes, I think, of a seminal paper in 2006 showing this more posterior predilection for white matter hyperintensities in the context of clinical AD. If we use baseline MRI data to predict who's going to get incident clinical Alzheimer's disease in the future, what we see again is a specificity of the parietal lobe white matter hyperintensity burden predicting incident clinical Alzheimer's disease. We turned to genetic forms of white matter hyperintensities because when we submitted papers, everyone sort of said, well, you're studying mixed dementia, not Alzheimer's disease. So we started to think, well, what's the purest form of Alzheimer's disease? And we started to think about autosomal dominant Alzheimer's disease, like in the Diane study. So we published this paper led by Sanju Lee a few years ago, and what we showed was that individuals who had mutations for autosomal dominant Alzheimer's disease, mostly presenilin 1 and 2 mutations, had increased white matter hyperintensities up to 6 or 7 years prior to their expected symptom onset. And again, when we look regionally, there's not much of a story in frontal or temporal lobes, but parietal and occipital lobes, we see 7 years prior to symptom onset, this increase in white matter hyperintensity volume, and in the occipital lobes, up to 22 years prior to symptom onset, we see an increase in white matter hyperintensity in this genetically deterministic form of Alzheimer's disease. They're relatively young and they're relatively vascularly healthy, if that's a word. So we've been doing a lot of work more recently in the context of the Alzheimer's Biomarker Consortium for Down syndrome, ABCDS, another genetic form of Alzheimer's disease. What's very interesting, there are lots of things that are interesting, but one thing that's very interesting about people with Down syndrome is that they seem to be protected from vascular risk factors, from peripheral vascular risk factors like hypertension. Those are the determinants that we often think of as risk factors for white matter hyperintensities. So we thought in the context of relatively healthy peripheral vascular disease, do we also see MRI evidence of cerebrovascular disease in this group? And this is a paper led by Patrick Lau, who's in the audience, a really lovely paper where Patrick characterized a bunch of vascular lesions. Here we're talking about white matter hyperintensities, and looked across the AD continuum in people with Down syndrome, in people who we call cognitively stable, people with MCI, people with possible clinical Alzheimer's disease, and people with definite Alzheimer's disease. And what he sees, what he reported was what we've shown over and over in late onset Alzheimer's disease, that with increasing diagnosis of AD, there's increasing white matter hyperintensity volume, and again, this is very much a posterior story, parietal and occipital lobes primarily. So how or why are white matter hyperintensities linked to Alzheimer's disease? So if you believe Brickman et al., white matter hyperintensities and other vascular lesions on MRI, and therefore small vessel cerebrovascular disease, are a core feature of Alzheimer's disease and should be integrated in our pathogenic models, our treatment and prevention strategies, and our overall conceptualization of the disease. If you believe reviewer 2, who keeps haunting me with every paper that we submit, white matter hyperintensities in the context of Alzheimer's disease are merely a reflection of cerebral amyloid angiopathy and are just another manifestation of amyloid pathology in the AD brain. And there's another reviewer 2 out there, I'm sure you're in the audience, who says this: white matter hyperintensities in the context of AD don't reflect amyloid, don't reflect cerebrovascular disease, but rather reflect AD-related neurodegeneration, so-called Wallerian degeneration, and is just a manifestation of tau-related neurodegenerative changes of the AD brain. So very different views about what these white matter hyperintensities are. And these are just a couple of examples. This was a very, very beautiful paper by Shirzadi and colleagues who looked at people with and without microbleeds across a number of different cohorts, and microbleeds as an indicator of cerebral amyloid angiopathy. And they showed that there's increased white matter hyperintensities in people with microbleeds, and people with microbleeds have more progressive white matter hyperintensities over time. They also showed in the 4 different cohorts that they examined, or 3 different cohorts that they examined, that vascular risk factors in these AD cohorts are not associated with white matter hyperintensity volume, either in the DIAN study, in the ADNI study, or in the HAB study, and they concluded that white matter hyperintensities are more likely to be associated with AD intrinsic processes of parenchymal and vessel amyloidosis, and white matter hyperintensity volume in the context of AD, or autosomal dominant AD, and load should be considered as a possible reflection of CAA progression. So that's sort of the CAA reviewer 2 take on things. The Wallerian degeneration take, this is a highly cited paper by Mackley's, another beautiful study, who looked at postmortem tissue and characterized postmortem tissue as having lesion, white matter lesion, or normal appearing white matter. And they showed among the Alzheimer's patients, there was increased markers of cytoskeletal breakdown or Wallerian degeneration in the white matter, but not among the controls. So I find these two, the CAA Wallerian degeneration, is somewhat unsatisfying explanations based on anatomical distribution of white matter hyperintensities, the temporality of the emergence of white matter hyperintensities, and other vascular factors besides vessel disease per se could account for some of the links that we see between white matter hyperintensities and AD. So let's talk about the anatomical distribution. So this is a very typical T2 flare that we see in someone, in this case with Down syndrome and clinical Alzheimer's disease. You can see the confluent white matter hyperintensity volume around the ventricles. It seems to expand towards cortex. And then you can look at the Boston criteria for cerebral amyloid angiopathy and the link there between white matter hyperintensities and CAA. And these are typically more punctate. It's like birdshot throughout the cortex and not the confluent periventricular white matter hyperintensities that we're used to seeing in AD. We also, in this case in the DIAN study, showed that presenilin 1 mutation carriers with cerebral microbleeds had increased white matter hyperintensity volumes, but statistically, the relationship between mutation status and white matter hyperintensity volume is not mediated by microbleed status. So we don't see this mediation between the mutation and white matter hyperintensity volume that's accounted for by microbleed as a marker of cerebral amyloid angiopathy. When we look at the anatomical distribution, also, you can see here that this is a frequency map that Mohammed Al-Shiko in my lab generated that shows the frequencies of white matter hyperintensities in the context of Down syndrome. And you can see, again, they're predominantly in the periventricular zones, and as it progresses, it goes more towards cortex. And if you think about Wallerian degeneration, where tau pathology is, and what types of neurodegenerative changes we see in Alzheimer's disease, those are predominantly cortical. So you would think, or you would expect that if Wallerian degeneration is accounting for white matter hyperintensities, those hyperintensities would be distributed proximal to where we see the neurodegenerative changes. And in fact, those U fibers that are most closely linked to the cortex, where we see degenerative changes, are particularly spared when we see white matter hyperintensities in aging in Alzheimer's disease. So we've done some experimental work to show that, in fact, supravascular disease causes white matter damage, as we all know, and that in turn promotes tau pathology, so the opposite of what we've been criticized for. So this is work led by Crystal Lang several years ago, where we used a transient occlusion model of the MCA and then reperfused the animals and had a sham control, and we showed that occlusion of the MCA indeed induces white matter damage, but not the sham control. And interestingly, or importantly, that occlusion also induced tau hyperphosphorylation in the medial temporal lobe, but not in the sham controls. And these were all wild-type mice, not genetically programmed mice for Alzheimer's disease. In work led recently by Natalie Edwards, this was just published, or just accepted rather, Natalie also showed that statistically in Down syndrome that white matter hyperintensities seem to give rise to p-tau biomarker concentrations through GFAP or through astrocytosis, not vice versa. It doesn't appear that statistically at least p-tau is upstream from white matter hyperintensities in our data. And then finally, in terms of the temporality of the emergence of white matter hyperintensity and other vascular lesions, this is work that was just published from Patrick Lau again, who examined the inflection point in people with Down syndrome who are all eventually or mostly going to develop Alzheimer's disease, at what point they develop vascular lesions. And you can see here, global white matter hyperintensities inflect at about age 35, perivascular space is a little bit earlier, microbleeds at about age 36, and infarcts, frank infarcts in Down syndrome inflect at about age 31. And if you compare that to the inflection points that we see in Alzheimer's pathology, including plaques and tangles, which here emerge at about age 35 or age 39, the vascular lesions seem to emerge at about the same time or slightly earlier than amyloid and tau pathology, suggesting again that causality, it can't really be the opposite direction where those pathologies cause the white matter lesions that we're seeing. So if it's not vascular risk, if it's not CAA, or if it's not Wallerian degeneration, what is it? What's causing those white matter hyperintensities to emerge in the context of Alzheimer's disease? This was work done by an undergraduate student, Famita Moni, a few years ago, where she looked again in the Down syndrome data and did a proteomic association analysis and showed that a number of markers that seem to suggest angiogenesis and inflammation are associated with white matter hyperintensity volume among cognitively stable and MCI individuals with Alzheimer's, with Down syndrome. And as participants transition to the AD clinical state, it seems to be more associated with neurodegenerative changes. And again, Natalie's working on some very nice analyses looking at some of these vascular biomarkers and showing that placental growth factor as a marker of angiogenesis or blood-brain barrier dysfunction seems to be upstream from white matter hyperintensities, and which gives rise to GFAP and p-tau downstream. And we're examining a lot of these possibilities in collaboration with Liz Head and Lisi Flores Aguilar and Donna Wilcock, looking at fibrinogen leakage with astrocyte colocalization. So in conclusion, I think we have good evidence that AD in the context of Down syndrome, in the context of autosomal dominant mutations, in the context of late-onset Alzheimer's disease, that white matter hyperintensities appear to be a core feature, at least very prominent. It's not attributable solely to exposure to vascular risk factors, and it may not be due, the white matter disease may not be due to vessel disease per se, arteriosclerosis in this case, but may be mediated by what we're thinking of as sort of endogenous vascular factors that affect the neurovascular unit and blood-brain barrier dysfunction and inflammation. Vascular risk factors may exacerbate effects and the manifestations of cerebrovascular disease, but at least in our hands, there might not be necessary for cerebrovascular disease to emerge in the context of AD. You don't have to have hypertension to have a cerebrovascular component of AD pathogenesis. There's evidence that the association between white matter hyperintensities and AD is simply a reflection of amyloid and tau pathology, but I think that this is still circumstantial. We have a lot to do in this area, and I'm excited that there's an audience here who's also interested in this. And let's use these observations to probe new ways of conceptualizing AD rather than simply forcing these ubiquitous neuroimaging abnormalities into this sort of amyloid tau neurodegeneration framework. putting a square peg in a round hole, as you will. So where do we go from here? So I just want to quickly highlight that there's a lot to do with white matter hyperdensities. They're not uniform lesions. Mohammed Al-Shiko in our lab is working on some really cool feature detection where we can characterize these white matter lesions and the diversity and the heterogeneity within the signal and the connectivity among the voxels in unique ways. We'll be hearing a little bit more about similar types of things later, and I think that's one direction we can go in for sure. So with that, I'd like to thank many of my collaborators and friends, and you for your attention. Thanks so much. So I think we're going to hold off, unless someone has a burning question or a tomato or something. We can hold off on questions until the end of the session. I'm going to introduce our colleague, Antoine Garnier-Coussard, who I mentioned before from Lyon, France, and you met him before, so let's get him up on the screen. Thanks so much, everyone.
Hi, everyone. I'm very happy to be here and to present this talk. I'm Antoine Garnier-Coussard, a geriatrician in memory clinic in Lyon, in the south of France, as well as PhD candidate in the research team of Gaël Chetla in the French Normandy, in the northwest of France. I'm so sorry I can't be with you in person today. I'm in France, but I'm very, very happy to make this presentation in the virtual fashion. In my talk today, I would like to argue that not all of white matter hypersensitivities are of purely vascular origin, and I want to highlight the existence of AD-related white matter hypersensitivities. I don't have any specific financial relationships to disclose, but I am a local sub-investigator or principal investigator of several trials in the field of Alzheimer's disease. So, as you know, white matter hypersensitivities are very frequent in aging, manifesting as large areas of T2 or flare hyperintensities in brain MRI. These lesions are mainly secondary to cerebral small vessel disease and are associated with cognitive impairment. However, all white matter damages, regardless of their nature, can appear as white matter hypersensitivities on MRI, for example, in inflammatory disease such as MS, in other inflammatory diseases such as neural lupus, in infectious, in encephalitis. In all these white matter damage, we can see white matter hypersensitivities on flare MRI. These lesions are not specific on a specific pathophysiology. The question arises, in AD, can white matter hypersensitivities be of non-vascular origin? To discuss this hypothesis, we published a perspective in Alzheimer's and dementia, compiling evidence for this idea. Today, I'm going to discuss 4 types of evidence supporting this hypothesis, and then future directions to test in vivo this hypothesis. The evidence comes from neuropathological study, from chronology of events, from topography of lesions, as well as from studies with genetics or biomarkers. Firstly, neuropathological studies highlight the heterogeneity of white matter hypersensitivities. Over 30 years ago, Lees and collaborators described cases of patients with leukorrhosis, synonymous of white matter hypersensitivities, without any vascular disorders. They hypothesized that white matter lesions are secondary to Wallerian degeneration triggered by Alzheimer's pathology, and not from a pure vascular origin. More recently, Kirsty McCauley and collaborators provided evidence for this hypothesis. They studied differences in the composition of white matter hypersensitivities from AD patients and controls, analyzing specific biochemical markers of Wallerian degeneration and hypoperfusion. They showed, as you can see on this slide, that white matter lesions in AD, and notably in the parietal lobe, are associated with Wallerian degeneration markers triggered by cortical AD pathology, whereas white matter hypersensitivities in controls are mainly due to ischemia and to cerebral small vessel disease. Secondly, white matter lesions are a very early event in Alzheimer's disease and appear independently of vascular risk factors, notably hypertension. In the study of the Diane cohort, carriers of autosomal dominant AD mutations showed an increase in white matter hypersensitivity volumes several years before symptom onset, and more than 10 years before for parietal and occipital white matter hypersensitivity. These participants are relatively young and lack specific vascular risk factors, suggesting a non-vascular origin for these lesions. In this study, our group also showed increased white matter hypersensitivities independently of vascular risk factors in Alzheimer's disease. For that, we included 54 AD patients and 40 healthy controls matched for vascular risk factors, so they did not differ in terms of age, in terms of gender, of systolic and diastolic blood pressure, as well as glycated hemoglobin. Patients had higher volumes of white matter hypersensitivity, as shown on this box plot displaying white matter lesion volume according to diagnostic group and brain regions. This higher volume was found in all brain regions, with the greatest effect in the parietal lobe. Moreover, these parietal lesions strongly correlated with lower cognition. Recently, in this study, Shirzadi and collaborators showed in this paper published last year in JAMA Neurology that increased white matter hypersensitive volume in AD is associated with neurodegeneration, with amyloidosis, vessel amyloidosis, and parenchymal amyloidosis, but not with elevated systemic vascular risk. They also show that increased white matter lesion volume may represent an early sign of CAA, cerebral amyloid angiopathy, preceding the emergence of cerebral microbleeds. Thirdly, the topography of lesions had more evidence in favor of the hypothesis of non-vascular white matter hypersensitivity. White matter lesion topography globally mirrors patterns of neurodegeneration. For example, white matter lesions are more anterior in frontotemporal dementia, more posterior in PCA, posterior cortical atrophy in typical AD and in dementia with Lewy bodies, and more left lateralized in logopenic progressive aphasia. So, as you can see, that globally mirrors pattern of neurodegeneration. Of course, neurodegeneration may also be secondary to cerebrovascular lesions, as brain atrophy is a major finding in cerebral small vessel disease. But in this focal neurodegenerative disease, frontotemporal dementia, logopenic aphasia, PCA, neurodegeneration is more likely secondary to specific proteinopathies and not to cerebral small vessel disease. Fourthly, white matter lesion and AD share some genetic risk factors. For example, posterior white matter lesions are associated with family history of AD, with ApoE4 genotype, and with AD polygenic risk score. Moreover, GWAS studies have also found common loci for AD and for white matter hypersensitivities in older adults. In terms of relationships between white matter lesions and AD biomarkers, white matter hypersensitivities are generally associated with amyloid biomarkers, but the relationships with tau biomarkers remain quite unclear. We propose a model illustrating the heterogeneity of white matter hypersensitivities. On the left, in green, you can see there is Alzheimer's disease. On the right, with red color, vascular disease. In the middle, a large overlap representing mixed disease or mixed dementia. White matter hypersensitivities appear at the boundary between these two entities, with a large proportion caused by vascular lesions, since vascular origin is the main factor explaining white matter lesions. However, there are also lesions in the overlap between AD and vascular lesion, which may correspond to white matter hypersensitivity secondary to cerebral amyloid angiopathy. And finally, there are also purely AD-related lesions, not of vascular origin, likely related to specific neurodegeneration or neuroinflammation seen in AD. The future step will be to create a lesion identity card for each patient, for each lesion, by disentangling the origin of white matter hypersensitivities, including vascular and non-vascular parts, as well as other mechanisms not discussed today, such as traumatic brain injuries, inflammatory diseases, etc., etc. At the individual level, we need to disentangle the pathophysiological pathway for each lesion. We could move from this approach today, an approach where all lesions are considered white matter hypersensitivities in the same way, to a more personalized approach where each lesion is interpreted individually. I think this is of major clinical importance. For example, if lesions are due to CAA, can we prescribe anti-amyloid drugs and lecanemab with the potential risk of amyloid-related imaging abnormalities are higher? If lesions are secondary to neurodegeneration, should we interpret them as an N biomarker instead of V biomarkers? If inflammation is involved, should we target inflammation and microglial activation as therapeutic strategies? Finally, if white matter lesions are secondary to sporadic small vessel disease, should we intensify hypertension and vascular risk factors management? Now, the questions remain, is it possible to highlight in vivo the heterogeneity of white matter hypersensitivity pathophysiology? We hypothesize that the multimodal neuroimaging signal within white matter lesion can help to understand the underlying mechanism. To do this, we extracted the intra-white matter hypersensitivity signal from T1 and T2 MRI, as well as intra-white matter lesion from FDG and amyloid PET in 142 participants in the Alzheimer's continuum. For example, we showed, as you can see in this plot on the left, that T1 and T2 signal within parietal white matter lesion are associated with amyloid status. Amyloid positive participants had higher T1 and lower T2 signal within parietal white matter hypersensitivity. Moreover, this intra-white matter hypersensitivity MRI signal correlated with cortical load quantified by floor beta P as UVR. Additionally, we attempted to classify each lesion using a clustering algorithm based on multimodal neuroimaging signal within a white matter lesion. This work is still ongoing and is still in progress, and I won't go into too much detail today, especially as Masha Dada will now explain these imaging approaches in detail for better phenotyping of white matter hypersensitivity. To conclude, I wanted to highlight that while white matter hypersensitivities are mainly due to cerebral small vessel disease in aging, they may also be secondary to other mechanisms, especially in Alzheimer's disease. This AD-related white matter lesion may be secondary to neurodegeneration, inflammation, and/or cerebral amyloid angiopathy, of course. We think that a more accurate characterization of white matter hypersensitivities in vivo could be very useful in clinical practice to improve precision methods. For example, should we avoid anti-amyloid monoclonal antibodies if white matter lesions are secondary to amyloid angiopathy? Should we intensify vascular risk factors management if white matter lesions are secondary to small vessel disease, sporadic and age-related small vessel disease? Should we interpret white matter lesion as a N biomarkers or a V biomarkers? Maybe it depends the lesion and the patients. And these questions have not yet been formally answered and need to be the subject of future study, which I believe will be very fascinating. Thank you all for your attention, and I thank all the researchers involved in the team, notably Gaël Chetla and all the team in Caen, in Normandy, and all my French colleagues, and as well as the local team, clinical research and neuroimaging team in Lyon, notably François Coton and all my university hospital in the Hospice Civil de Lyon. Thank you again, and I will be more than happy to discuss in further detail of this very interesting topic. I hope it has interested you.
Thanks so much, Antoine. Our next speaker is Masa Dadar.
Yes. Thank you. Thank you for the introduction. Okay. Okay. So in the previous talks, we saw that the white matter hyperintensities that we see in different patients and also different lesions in the same patient might not correspond to the same underlying etiology. What I'm going to talk a bit about today is whether we can use information from multimodal neuroimaging methods to get at the underlying etiologies of white matter hyperintensities in vivo. So based on postmortem studies, we know that there are a number of different underlying pathologies that can result in the hyperintense signals that we see on T2 weighted and FLAIR images. And basically, post-mortem, what we can do is get the white matter hyperintensity lesion tissue and run a series of histology and immunohistochemistry assays, for example, stain for myelin or astrocytes, to have a better picture of what are the underlying pathologies that we are associated with each specific lesion. So based on these types of studies, we know that white matter hyperintensities in different populations have been linked to demyelination, as axonal loss, edema, gliosis, arteriolosclerosis, and Wallerian degeneration with different levels of severity. If we try to segregate the results of postmortem studies a bit, we do start to see some patterns where white matter hyperintensities in small vessel disease patients are more reported to be linked to arteriolosclerosis, for example, whereas in Alzheimer's disease patients, there have been reports on relationship between white matter hyperintensities and Wallerian degeneration and more neurodegeneration-related processes. But this is, again, first of all, there's a lot of overlap between the pathologies that are reported to be associated with white matter hyperintensities in different disorders, and also white matter hyperintensities in the same patient in different brain locations can be linked to different pathologies. As was mentioned before, frontal white matter hyperintensities are supposedly more vascular, versus posterior white matter hyperintensities might be linked more to Wallerian degeneration. And again, if we think back on the findings of large postmortem studies, we know that up to 70-75% of Alzheimer's disease patients do have high levels of comorbid vascular disease. So it does make sense that they would have at least a number of vascular-related lesions, even if they have some neurodegeneration white matter hyperintensities. So back to in vivo, T2 weighted and FLAIR images allow us to quantify white matter hyperintensities and assess their volume. So if you look at these images, for example, we would be able to tell that the frontotemporal dementia patient has more white matter hyperintensities in the frontal lobe, whereas the Lewy body dementia patient has more posterior white matter hyperintensities. But on their own, the structural sequences might not provide us with enough information to get at the underlying etiology of the white matter hyperintensities. There have been some studies, including ours, that try to use the signal inside of the white matter hyperintensity lesions as a measure of severity and see if this adds some information above and beyond what we have with the white matter hyperintensity volumes. And we were able to show, for example, the white matter hyperintensity signal in T1 weighted, T2 weighted, or T1-T2 ratio, or FLAIR images, especially when it's normalized by the signal of the normal appearing white matter that is surrounding it in multimodal and multicenter studies, can actually relate to clinical conversion, for example, in Alzheimer's disease, above and beyond what the volumetric information of white matter hyperintensities can provide. So taking this idea of using the signal inside of the white matter hyperintensity lesions and combining it with the idea of running a series of different assays post-mortem to have a better picture of what the pathology of the lesions are, we can try to use multimodal MRI sequences information to have a better picture of what the underlying etiology of the white matter hyperintensities are in vivo. So two imaging modalities that would be particularly suitable for this type of analysis would be diffusion-weighted imaging-based measures and quantitative MRIs, since they are linked to microstructural and biophysical tissue properties. At a course level, if we want to categorize them, mean diffusivity and ISOVF measures are sensitive to water content in the brain. FA, ICVF, and OD are sensitive to health or damage to the fibers. And then quantitative T2 star and QSM images can be sensitive to myelin and iron. So when we put the information from these multimodal MRI sequences, we might actually be able to get at some of the underlying etiology of the white matter hyperintensities in vivo. So this is the work that mostly Olivier Parent has been doing during his PhD. If anyone is interested, I really encourage you to check out his poster on Tuesday. He's much better at explaining his work, of course, than me. But basically what Olivia has been trying to do is to use the UK Biobank data that has all of the imaging modalities that we need to extract the multimodal image informations that I talked about in the previous slide to look at the white matter hyperintensities. So he segmented the white matter hyperintensities using T1 and FLAIR images from the UK Biobank and an automated segmentation tool that was trained on UK Biobank data. And he also generated custom UK Biobank-based templates for each of the imaging modalities that he was using to improve his data processing and normalization. He then generated normative models of what healthy tissue microstructure looks like for each of these different 7 MRI features at each voxel level that were agent sex appropriate. So if we look on the left, for example, the blue dots are the values of MD for 2 voxels in the anterior and posterior regions, and from their comparisons against the normative models, we can see that, for example, MD in the anterior region is more abnormal than the posterior region, whereas it's the opposite for FA. So if we put this type of information together, we might have a better idea of what the underlying etiology is for each specific lesion. So using these normative models, Olivier then generated Z-score maps at the voxel level for each of the 7 imaging modalities, and then we can put together information from all of these Z-score maps to see for each individual where the specific abnormalities are at the voxel level and for each imaging modality. So the first question that we wanted to answer was, is the signal in these 7 different sequences different enough that we would extract patterns of abnormality in a specific white matter hyperintensity related clusters? So using a completely data-driven approach, we had 3 clusters, 1 periventricular, 1 mainly posterior, and 1 mainly anterior, which makes sense basically based on what we talked about from postmortem studies and what you heard earlier today. So then Olivier looked at the Z-score values in these 3 clusters and 7 measures in 4 different disease populations in UK Biobank. 3 of them were vascular, and 1 of them was a group of neurodegenerative dementia patients. And basically what we found interesting was that the patterns of the abnormality in each of these measures was not the same. So for example, if you look at the FA, it has more abnormalities in the anterior regions in the stroke group, whereas the OD has more posterior cluster abnormality in the dementia patients. So this might allow us to differentiate the type of white matter hyperintensities a bit. So all of those results were at a group level. The next question that we wanted to ask was whether we can do this type of analysis at an individual level. So an ideal study design, if you want to look at this type of analysis, would be, as was mentioned previously, to have a data set that has multimodal MRI information in vivo, and then we would also have postmortem pathology for each of the individual lesions, and then we would try to see if we can predict postmortem pathology of the lesion using these multimodal MRI sequences as features. But of course, we didn't have this information in the UK Biobank, so we tried to do something at a much more coarse level, which was to predict future disease diagnosis just based on the signal inside of the white matter hyperintensity lesions. So we had 15 participants in the UK Biobank data that did not have any diagnosis of neurodegenerative dementias at their baseline, but were diagnosed with Alzheimer's disease at their follow-up on average 3 years from the baseline MRI visits. So we matched these participants with 95 individuals that had a diagnosis of vascular disease around the same time of follow-up. Using a machine learning framework, we were able to predict future diagnosis of these participants with a balanced accuracy of 64% just based on the white matter hyperintensity signals. I want to emphasize here that the idea is not to have an excellent future prediction in disease classification accuracy. The idea is that the white matter hyperintensity signal in these multimodal MRI features was different enough that it could actually accurately predict up to 64% accuracy, the future diagnosis of these patients. And again, since we also know that, for example, in Alzheimer's disease patients, we will probably have a lot of vascular lesions, lumping all of them together and having one diagnosis would really reduce our prediction accuracy. So along the same lines, we did another classification analysis. This time we took 100 participants with the top polygenic risk scores for Alzheimer's disease versus the 100 participants with the top polygenic risk scores for vascular disease, and we tried to use white matter hyperintensity signals to predict the diagnosis for these patients. And again, we were able to have a balanced accuracy of 63%. And for both of these classification tasks, another thing that was interesting was that the most significant features that contributed to accuracy of the classification were, again, OD and FA. So they might actually have enough information that differentiate the vascular versus AD-related lesions. So to summarize all of these points, the multimodal white matter MRI signal in white matter hyperintensities shows that the expected location patterns that we know from postmortem studies, and might actually provide enough information that would allow us to differentiate between AD and vascular-related lesions at an individual level. I want to thank my team, especially Olivia, whose work I presented today. Thank you.
Thanks so much. That was a great presentation. We have one more talk from Andreas Haridimu from Boston University. If we could just load up his...
Good afternoon, everyone, and thank you so much for joining this dedicated session on wide matter hyperintensities and cerebral amyloid angiopathy in Alzheimer's disease. I hope it's becoming very obvious throughout the course of the other talks that wide matter hyperintensities are extremely heterogeneous from both a pathophysiological point of view and from a clinical point of view. However, in my talk, I will specifically focus on cerebral amyloid angiopathy, and instead of taking a pathophysiological and theoretical perspective, I would like to just target a couple of key practical clinical applications on how wide matter hyperintensities can inform the diagnosis of CA within the memory clinic patient population. And without further ado, as an introduction, cerebral amyloid angiopathy is defined at the pathophysiological and pathological level as gradual amyloid beta protein accumulation within the cortical leptomeningeal small vessels. And this is a gradual process which takes years to affect the vessels, both physiologically and from a structural point of view. CA is an extremely common pathology in the brain, especially in patients with Alzheimer's disease, as these pointers show here. These are basically a meta-analysis of all the pathologically based data, which looked at various populations, in this case, patients with a pathological diagnosis of Alzheimer's disease, and they look at the prevalence of CAA. In patients with Alzheimer's disease, any CA presence of any severity is found in more than 80% of these brains, whereas moderate to severe CA is found in about 50% of patients with Alzheimer's disease. Why is this relevant? Apart from how common this pathology is, cerebral amyloid angiopathy has specific clinical relevance. The easiest way to define this in the stroke field is that CA is associated with high future risk of lobar hemorrhage and hence has huge implications for oral anticoagulation decisions in atrial fibrillation. In the memory clinic and in Alzheimer's disease, the clinical relevance is that CA is a key vascular contributor to cognitive impairment and dementia, even in patients with a diagnosis of Alzheimer's disease. In the last 1 to 2 years, CA has emerged in clinical relevance because it's the most significant risk factor or pathological underlying of ARIA, amyloid-related imaging abnormalities. And in one sense, ARIA is essentially iatrogenic CA-related inflammation. Hence, a diagnosis of CA, an accurate diagnosis of CA, has huge implications in the selection of patients for anti-amyloid treatments like lecanemab. And again, I just want to spend a couple of minutes on this concept and highlight in a simple and rather superficial way that ARIA, which occurs as edema or hemorrhage in some patients who are on anti-amyloid treatments, is essentially an iatrogenic form of CA-related inflammation. And as you can see in a rather dramatic example of a patient who was on lecanemab and he was given TPA for presumed ischemic stroke, in extreme forms, this syndrome can be manifest in multiple superficial hemorrhages and multiple areas of edema. And we know from limited but quite well-performed neuropathological studies that pathologically, these manifestations correspond to cerebral amyloid angiopathy. You can see the amyloid here in brown, with destruction of the vessel and associated inflammatory infiltrates. Essentially, anti-amyloid treatments in selected patients who presumably have some underlying CA leads to acceleration of this CA pathology in addition to inflammation. And essentially, both from a pathological point of view, clinical point of view, and neuroimaging point of view, this syndrome is essentially identical to what is known as CA-related inflammation. Now, moving on to diagnosis. The diagnosis of CA is pretty easy when the patient presents with hemorrhagic manifestations, so a lobar hemorrhage with associated lobar microbleeds or extensive superficial siderosis. And it's basically a spot diagnosis in a way, and I'm sure you're all familiar with similar images. If these images are very familiar and you think they look like CA, you have essentially used the Boston criteria. In their initial forms, the Boston criteria were established to assign the probability of a lobar hemorrhage being due to CAA, and they have evolved over the years to be more inclusive. The previous version of the Boston criteria, called Boston criteria version 1.5, they defined probable CA, which is a category most useful in clinical practice because it gives the best diagnostic accuracy without the need for neuropathology. It is defined as the presence of multiple, 2 or more bleeds, macrobleeds or microbleeds, or a single bleed, again, macrobleed or microbleed, and any cortical superficial siderosis. All the hemorrhagic manifestations, they need to be on strictly lobar locations, so no deep bleeds or microbleeds are allowed. And of course, no other cause of these hemorrhagic manifestations should be evident. These criteria, they were validated basically in a handful of cases, mainly coming from hemorrhagic stroke patients. And until recently, they haven't been expanded to include the whole spectrum of CA imaging manifestations. Just quickly to mention that the Boston criteria are so well established and successful because they're based on the underlying topography of CA, which, as we saw, is a very superficial lobar pathology, and hence the hemorrhagic manifestations, they follow this topography, versus non-amyloid small vessel disease, which primarily affect the deep perforators, so lenticular striatal arteries, or perforators from the basilar arteries. And when they affect more superficial cortical penetrators, they affect, again, the longer penetrators. Hence, the manifestations that result from non-amyloid small vessel disease can be deep hemorrhages, deep microbleeds, or sometimes mixed. But when the microbleeds within non-amyloid small vessel disease are lobar, they're a bit deeper compared to CAA. We have a new version of the Boston criteria, which they were validated about 2 years ago. And now they include the whole spectrum, essentially, of CA MRI markers, both hemorrhagic markers, which I'm not going to cover here, but also non-hemorrhagic MRI markers. And the 2 main MRI markers, which are non-hemorrhagic, and they emerged with multiple lines of evidence as supporting features of CA, are severe perivascular spaces in the centrum semiovale, which are best identified on T2 sequences. They have the same intensity as the CSF. And when there are 20 or more in any given hemisphere, single cut, they count towards the Boston criteria for CA diagnosis. The other non-hemorrhagic manifestations is what we call multiple subcortical spot white matter hyperintensity pattern. And it's a specific pattern of white matter hyperintensities, which is cortical or cortical subcortical. It essentially, as the name implies, consists of circular or oval spots. And when there are 10 or more in the cerebral white matter, it seems to be a good supporting feature of CA in the right clinical context. Just a bit more details about these spots. They're best assessed as other white matter hyperintensities on FLAIR images. Essentially, you look throughout the brain in the cortex and subcortical areas. You look at all slices, both hemispheres, and the foci are counted as such if they're restricted to subcortical areas close to the cortex, and they should be isolated. So if they start to confluent or they're continuous with periventricular white matter fiber intensities, we don't count them as part of this pattern. The same applies if these foci are related to acute or chronic infarcts, lacune, large perivascular spaces, or intracerebral hemorrhage. These do not count into this pattern. There's no established size limit, but most of them should be visible without windowing extensively your images and without using any magnification. And again, if there is confluence, many of these lesions merging together, creating larger patches, they shouldn't be counted as such. As soon as there are 10 or more subcortical white matter fiber intensity foci fulfilling this criteria, we define this as the multispot pattern, and they count towards the Boston criteria. You see here, as an example, multiple discrete spots of white matter fiber intensities. This is the same patient. Again, this is some encephalomalacia, which is not taken into account. These areas here, which are merging, are not counted again. Periventricular areas are not part of this pattern. So in this example, there are at least 10 independent small foci in subcortical or superficial areas. So hence, this is counted as the pattern being present, and this is a supporting feature for a CA diagnosis. Based on the Boston criteria version 2, probable CA is now defined as not only having 2 or more strictly lobar hemorrhagic lesions in any combination, but also as having 1 strictly lobar hemorrhagic lesion plus one of the white matter features. For example, a patient with a single superficial microbleed and presence of the white matter subcortical spot pattern can now be diagnosed with probable CA. And this is the patient population that we commonly encounter in a memory clinic. Most of those patients, because they do not have the advanced hemorrhagic subtype of CAA, they do not present with florid lobar hemorrhages or hemorrhagic manifestations, but they rather have these non-hemorrhagic features of CAA. The best performance of the Boston criteria based on a multi-center validation study we have performed. Again, we're looking at the combination of the criteria I showed you for probable CA against the absolute gold standard being autopsy. As you can see here, sensitivity is about 75% and specificity is 95%. And this diagnostic accuracy is excellent in patients presenting with hemorrhagic stroke, above 90% for both. However, in patients similar to those that they come to the memory clinic with non-hemorrhagic stroke, while the specificity is quite high, the sensitivity is low, it's about 55%. And of course, in both scenarios, these criteria are much better than the prior version of the criteria, which did not include any non-hemorrhagic manifestations. What does this mean in practice? This means that when there is a patient, let's say with Alzheimer's disease diagnosis, who fulfills probable CA criteria, there is high degree of certainty that this patient indeed has some, at least moderate to severe CA pathology. However, if a patient does not fulfill the criteria, we cannot conclude with high degree of certainty that the patient does not have CAA. In other words, these criteria are good when when they are fulfilled for probable CA. However, we're missing a lot of patients who do not currently fulfill the criteria, but still might have significant CA. And those are the patients that we need to capture better, especially in the era of anti-amyloid treatments. Now, CA being a small vessel disease pathology is invariably associated with white matter fiber intensities in general, including periventricular white matter fiber intensities. So over the years, there have been efforts to look whether a specific topographical pattern of these periventricular fiber intensities with a focus on posterior areas might be an additional marker of CA. And the idea behind looking at this topographical pattern is that we know for many years from neuropathological studies that for whatever reason, both leptomeningeal and intracerebral CA is more severe in the occipital cortex and in the posterior cortical areas in general. And then, of course, it will go to expand and affect more frontal areas. But in many patients, the posterior areas will be more severely affected. As you can see in this example, this is a patient with CA from a memory clinic, and it's very obvious visually that the posterior periventricular areas are more severely affected than the frontal areas. Whereas this is a patient with non-CA small vessel disease. The patient had lacunes and some deep areas of small vessel damage. And you can see that the frontal periventricular areas are obviously more severely affected compared to the posterior ones. Hence, there have been studies that they showed indeed that using semi-quantitative measures, severe posterior periventricular white matter cover intensities and or a higher posterior to anterior ratio might be an additional feature that might help distinguish these patients. However, in most studies, either the method was too complicated to use clinically, or the patients that showed this difference were already diagnosed with probable CA based on the previous criteria. Hence, there is room to look into white matter hyperintensity anatomical patterns in patients within a memory clinic that do not fulfill otherwise probable CA criteria. Now, the issue is very complicated, as we saw in other talks, because of coexisting pathologies. In most of the elderly patients, there might be coexistence of both Alzheimer's disease and downstream neurodegeneration, cerebral amyloid angiopathy, or non-amyloid small vessel disease. And any given patient might either have primarily severe CA with some Alzheimer type pathology, or primarily non-amyloid small vessel disease with Alzheimer type pathology and some of CA. Hence, there is a whole spectrum of these imaging markers that they overlap as indicators of all of these three pathologies. And this complicates the search for other amyloid markers in the CSF or radiological markers using advanced imaging, amyloid PET or tau PET, with a specific signature, topographical signature, when you have a primarily small vessel disease co-pathology. So for now, we have the Boston criteria, which they should be applied in patients older than 50 years, ideally using 3 Tesla machines on susceptibility-weighted imaging. And they do provide, I believe, a very nice conceptual framework on how to use current knowledge on MRI markers for a CA diagnosis. And of course, it goes without saying that it needs good familiarity with these component CA markers. The rater should be familiar with what the microbleed is, what the mimic is, what is this white matter cover intensity pattern that we'll talk about. The presence and the multiplicity of superficial hemorrhagic markers are probably the best diagnostic markers for CA currently. However, especially in the memory clinic and in patients with Alzheimer's disease, the use of white matter features, which are now included in the Boston criteria, are going to prove to be extremely useful in classifying these patients and potentially preventing any downstream ARIA when anti-amyloid treatments are considered. The role of white matter cover intensities, I expect to be expanded in the future as a supporting feature of CA, especially in these non-hemorrhagic presentations. This might be the posterior distribution or any different combination of white matter cover intensities that is going to be validated as a good diagnostic tool. And we didn't have time to cover this, but the other MRI markers associated with a specific white matter cover intensity pattern might give us important clues for the presence of CA. For example, even when there is posterior predominant white matter cover intensities in a patient, let's say, with multiple deep lacunar infarcts and deep perivascular spaces, I would have low concern for CAA, whereas the presence of the same posterior pattern in a patient with, let's say, severe white matter perivascular spaces or with a single lobar microbleed will increase my pretest and post-test probability of the patient actually having CA. So we need a really comprehensive framework when we look at these MRI markers, and we often look at the company they travel with in order to tease apart the relative contribution of small vessel pathologies and hence infer how likely CA is going to be in these patients. Thank you so much for your attention.
That was great. Thanks to all the speakers. We have a number of questions online, but why don't we open it up for the audience? And as you're running, hey, Prashanthi. Oh.
Hi, Prashanthi Vemuri, Mayo Clinic, Rochester. Great talks. 4 of you did a great job presenting what's at the cutting edge of WMH. I have many questions, but just limit to 2. First one for Adam. So I agree with your results on early onset AD in the Diane cohort and Down syndrome. We see the same thing with leads. WMH seems to go up with disease severity, but I differ on your opinion with LOAD. The reason is that you see a lot of elderly with WMH, no amyloid tau for years, And the other most important reason is in the ADRC cohorts, if you looked at all the WMH, not every AD participant has significant WMH. Very small proportions have high. So if WMH was a core feature of AD, how would you explain that?
I've yet to see data from an AD patient that doesn't have any white matter disease, but
Everybody has little. I'm saying that if it's a core feature, it should be associated with disease severity, and the extent should be sufficient where you say it's significant.
yeah, I don't think we have a good handle on what the amount of white matter hyperintensity is sufficient to cause a link between with AD symptomatology or pathogenesis. So it's a good question. It's a valid question. But I suspect that as we think about all the different contributors to AD, we can think about sort of like what Andreas had in one of his last slides, that there are different degrees of contribution of different pathologies to AD. But I don't think that there are any pathological series or many AD MRI series that would suggest that there's an absence of vascular involvement. And if there is, they're not looking hard enough.
Thank you.
That's my guess.
Quick second question. It's based on what you said. For the second and third speakers, great talk. If we do not know the amount of AD that is needed to cause WMH changes, how could you use WMH to pinpoint to the etiology? So the question was that you said by looking at WMH, you could probably pinpoint to the etiology, but it's so heterogeneous. How could we do that? Like, how could we pinpoint to the etiology?
Yeah.
Yes, it is very heterogeneous. And basically, I think the ideal scenario, if you want to look at this, would be to have postmortem pathology assessments for each of the specific lesions and then try to put together a picture to see, okay, how much of them are from what etiology. But at the in vivo level, there is a difference in the signal that we are getting. It might be that we get lucky and it's actually like one of the major neuropathologies that is associated with white matter hyperintensities, but it might also be a third factor that is causing this. We won't know until we actually do the lesion-specific at the individual level analysis to link to what the signal that we are getting is. So, of course, I talked about like there are differences, but we won't know until we get there, and I don't think we are, at least our group is not there yet. We are starting to look at the postmortem level, but I think in general, based on the studies that are coming out postmortem, is you can't really say white matter hyperintensities in this person are based on this pathology. Each person is going to have a mix of lesions that come from different etiologies. That's what the second speaker, is he connected?
Yeah, can we get the speaker?
Yes, I'm here.
I think
okay.
he will have, yes. Yes.
Can you hear?
Yes, I
Hi,
think
can
you
you hear
should
me?
take yes.
Yeah, I think it's a great question and it's very hard to answer. But we think that future study will move from the individual level to the lesion level and to accept that one individual may have different types of lesions and not all of lesions in one brain are from the same origin. So maybe including in future study, including in statistical models and in different mathematical approach, we'll move from one individual to one lesion and to study each lesion individually, maybe to accept this heterogeneity. And I think we don't have enough results to promote this approach, but I think the approach from the team of Masadada with multimodal neuroimaging within lesions is a very good approach and very relevant, and I hope it will help to disentangle the etiology of each lesion.
Thank you.
Why don't we take a question here?
Hi, Jie Kang from Johns Hopkins University. So I have a question for the Dr. Brickman. So you are trying to link the white matter hyperintensity with Alzheimer's disease. But for the Alzheimer's disease patients, it seems like the postmenopausal woman has higher possibility to get Alzheimer's disease. Do you have seen any like a sexual difference for the white matter hyperintensity?
It's a great question. I haven't looked too deeply. Some of my colleagues I know have, but I off the top of my head, I couldn't describe to you the sex or gender differences that we see. But sometimes they emerge in some of our analyses, other times they don't. I don't know if any of my colleagues have strong findings. We're going to look into it.
Yeah, thank you.
In the back there.
Yeah, hi, thank you. Great session. My name's Tammy Bensinger. I'm at Washington University in St. Louis. My question stems from the last presentation from Dr. Chardemou, but is actually open to any of the panelists. So his comment was that ARIA is CAA-RI, so the form of CAA that has the reactive inflammation with it. I've actually heard other people today in other sessions saying a goal of management of early mild ARIA would be to consider giving steroids to prevent it progressing to a CAA-RI type picture. And I also want to point out, when you look at the clinical trials where we have placebo people and it's reported as an ARIA rate, that's actually just CAA, and I don't think that's CAA-RI. And so my question to the group is, I don't think any of us has a good answer for this, but what types of experiments or projects do we need to think about doing in order to better distinguish between CAA that's spontaneous in the setting of a therapy and CAA that is actually ARIA in a therapy? Because I think we see both of those happening.
That's a great question, Tammy. I'm going to ask Andreas to come on the screen if he...
I think, unfortunately, Andreas is not connected.
It's a great question, and I'm sure Andreas would have very profound thoughts about it. I'll take a stab. I think looking at baseline predictors of incident microbleeds in the placebo group is going to be very, very important to establish sort of base rates of what we would expect in a clinical trial. For microbleeds, but microbleeds are certainly part of Alzheimer's disease and certainly seem to be related to treatment. So I think that more analysis of clinical trial data, and that's been a repeated theme today, I know, but that seems to me the most logical approach to trying to...
I just have this hope if we could figure that out, we could really better identify the people that would get severe ARIA. Because right now we're just calling it all the same.
I think that's a great point, and I think with enrollment in clinical trials, with the focus, not exclusively, but primarily on baseline microbleeds as a risk factor, I think there are a lot of things we could look at in the baseline MRI data across modalities to predict some of those adverse outcomes versus what's just the normal course of the disease in the placebo group. I don't know if my colleagues have other comments. No. Okay.
I just wanted to add that in phase 3 clinical trials, people with probable CAA were not included as extensive and large white matter hyperintensities were systematically an exclusion criteria, so we don't have information about the links between large white matter lesions and risk of future ARIA, and people with more than 4 microbleeds were also not included in these trials. But the question for steroids, I'm not in Philadelphia, so I didn't hear this previous session today. I think it's for the case of area with edema and not with hemorrhagic features. Can you precise the question with...
So the question was whether to administer steroids in order to prevent CAA-RI.
Okay. Including without edema, not only the edematous form?
I'm not sure. Tammy, do you want to clarify? Or-
Okay.
Well, we can move on, and maybe Tammy
Yeah, okay.
offline we can have. And all the way in the back? Yeah.
Wonderful session. Thank you. I'm Paul Yushkovich from UPenn, and my question is mainly for Dr. Dadar. So it's amazing work with the different modalities, and you stress the need to validate in postmortem imaging. But I think one difficulty is finding postmortem data sets where you would have all of those modalities. And so my question is, have you looked at whether there's some minimal set of modalities where you could still infer some of those patterns and some of those classifications without having all of them?
That is a great question. First of all, we are doing the postmortem work. At the Douglas Research Center, we do have a brain bank of more than 3,000 patients with brains with different neurodegenerative disorders. And we are doing all of the multimodal imaging MRI that I talked about. And then the idea is to extract the white matter hyperintensities and do all of the assays to try to link them. So hopefully in future that will be a thing. But basically what the data that I presented today was the minimal set of node-based measures, and the quantitative MRI signals are still a bit in debate between us, at least. One of the other things that I would like to emphasize is that we will be trying to replicate the results in ADNI and other data sets. We would like to try to do the same type of analysis in multi-center data sets to see how stable and robust these white matter hyperintensity signal features are between data from different scanners, different centers, and different sequences. All of these validations, of course, are necessary for us to get there. But I would say T2 star minimum and multi-tail diffusion. Yeah.
So we'll go up front, and then after that, Batul.
Hi, thanks. Neil Parikh, vascular neurologist at Alnylam Pharmaceuticals. Terrific session. Enjoyed having my assumptions about the vascular causes challenged. My question is, and I know Andreas is offline, but the posterior predilection of white matter hyperintensity lesions in CAA, can any of the speakers talk about the pathophysiology of that and how it could inform the understanding of white matter hyperintensities, generally speaking, in terms of the debate between vascular and non-vascular causes?
Andreas? Andreas is not on still, right? Antoine, do you? Yeah?
We miss Andreas for this kind of question.
Yeah.
I think from a neuropathological point of view, there is a predilection of leptomeningeal, very small arteries and very small vessels, and posterior vessels in CAA. But I can't explain the reason why. But maybe Adam or Masa can complete my answer.
Yeah, I mean, I think what we've been trying to do is combine some of the imaging markers with biofluidic biomarkers that hopefully help point to different mechanisms that might be involved with the different lesion types, regionality, and texture. So that's how we're approaching it. It's a lame answer, I know, but that's
if I might just make a quick comment.
Oh yeah, thank you. Yeah.
So I mean, it's a very good question for which the answer isn't completely known, but it's not just CAA that shows sort of this pattern. So there's reversible progressive leukoencephalopathy syndrome, RPLS, that also shows you the same phenomenon where you have this temporary and very robust posterior circulation white matter hyperintensity increase. And I think that the reasons for it aren't exactly known, but there's an unusual ability to auto-regulate vascular caliber within the CNS, and it appears to be a little bit different between the anterior and the posterior circulation. That's a little bit of a hand-waving answer, but it's not just unique to CAA.
Awesome. Thank you, Dr. Chatwell. Not quite yet, Dr. Batul Rizvi.
Sorry. I think my question is very similar to the one that was just asked, but it was basically asking whether there's, if the regional distribution of microbleeds from CAA is differentially related to downstream markers like tau and neurodegeneration. So, and I know Dr. Andreas is offline, so I'm not sure if other.
So we haven't looked at that. It's a great question. We don't see a whole lot of microbleeds to relate to some of these downstream factors. Have you guys looked at? No, and Antoine, have you looked at the association of regionality of microbleeds with downstream neurodegenerative and tau changes?
No, I didn't.
No, but it's a great idea, and I think there are a lot of data sets that we could probe that in. Yeah.
Hi, Meher Judikonda from MGH. I'm curious what you think about the role for hemodynamic imaging techniques in all of this, and where you think they could help answer some of these questions about hemodynamic insufficiency and other aspects.
Totally, 100%. I think the more vascular-related imaging modalities we can collect, I think the better, just to get a clearer picture of what we're measuring. Do you have other comments? There's someone, Owen, before you, is that okay? Unless you have a direct, all the way in the back?
Yes, thank you for the great session, Dr. Brickman. My question is, I'm thinking about animal models of AD where we try to mimic sporadic AD. Do we see white matter lesions or something similar in those animal models, or is at least anybody looking into that as well?
Yeah, I think there's a fair amount of work on oligodendrocyte abnormalities that emerge in the context of AD. Mice don't have a whole lot of white matter disease, or a whole lot of white matter, let alone white matter disease. But our approach was to induce white matter disease and see if it can promote pathology. But at the cellular level, certainly there's oligodendrocyte involvement and some of the sort of associated neurovascular unit type cells, endothelial cells, et cetera, and the inflammatory cells, inflammatory reactions interacting with those different cell types. So I think you could lawyer a narrative that connected what we see on human imaging with what the animal models are showing. Owen, yeah?
Yeah. So I think the short version of my question is, why are we still throwing around the concept of white matter lesions per se? Is it all for historical reasons? Because, I mean, I think from Massa's data and from Pauline Maillard's data from way back, it's clear that we're shutting off information that's potentially clinically useful by defining white matter hyperintensities in a yes or no fashion.
Yeah.
Even if you just look at the flare signal continuously, and especially with these other types of signals. So, why are we still talking about white matter lesions as opposed to these kind of more continuous features?
Yeah, it's a great point, and I think some of the more advanced quantification approaches are getting at that, the heterogeneity and certainly the dynamic aspects of the signal underneath white matter hyperintensity. It's a really, really good point. I think I tried to find the very first paper, and not surprisingly, I think it was Moni de Leon in the 80s who most people in this room maybe weren't alive. So it's been a while. It's been a while that we've been just doing this.
I just have a comment on the previous question. So you could see some white matter hyperintensities or ARIA-E-like or H-like lesions in some non-human primates. So we have a poster on Wednesday. I can talk to you later.
Thanks. Could you tell us who's presenting the poster? Or?
Yes, the gentleman over there, Suleiman.
Okay.
What is the number of our poster? I apologize.
Okay.
It's on a squirrel monkey model, so we are also correlating the lesions to biomarkers in blood and CSF.
Okay, thanks so much. There are a number of questions online. I can try to read them. This is for Antoine. What is the longitudinal progression of white matter hyperintensities? Is it different or has different trajectory in AD-related white matter hyperintensities versus white matter hyperintensities of vascular origin?
To the best of my knowledge, there is no data that can precisely answer to this question. But my impression is that the trajectories of AD-related or vascular-related lesions are quite similar, We know that vascular-related white matter hypertensities can be very stable with time, and some of them can increase and some of them can decrease across time, but we don't have the longitudinal information for AD-related white matter lesions, and it's hard in studies to confirm that lesions are AD-related and not vascular-related. So today we can't answer precisely to this question, but it's a very important and good question.
I can add on that I think we have looked in the ADNI data set separating the white matter hyperintensities progressing in amyloid positive and amyloid negative groups, and the amyloid positive groups had faster progression, significantly faster progression of white matter hyperintensities. And also converters versus non-converters. So amyloid positive converters progress faster, and amyloid positive in general progressed faster than amyloid negative. I think we had like 4 or 5 years of follow-up on the white matter hyperintensities.
Go ahead.
So, Ahmed Bahrain, University of Kentucky. I have one comment that question that asked previously, so I hope I'm not late. So I ran 6 differences for white matter hyperintensity. I saw female, small sample, so we need to do more investigation about that. So I saw the female has larger volume white matter hyperintensity compared to the male, but still, small sample size, we need to do more investigation about this.
Thank you for that context.
I can comment on that a bit as well. I feel like the 6 differences in white matter hyperintensities is very cohort dependent. We have looked in cohorts, we haven't had any different results. We have had higher volumes in males versus females, females versus males. I think the age range of the population and the risk factors are very important when we look at white matter hyperintensities and sex, and we need to consider these factors in larger samples to know better.
Let's try to get 2 more quick questions in the back, and then Jasmine after. Yeah.
Hi, Rin Flaherty from New York University. So we seem to be talking about vascular versus AD in this very binary way in this panel, but there's a fair amount of evidence that there's neurovascular disruption in Alzheimer's disease. Yeah. So how do you tease that apart?
I think that's sort of my point. Like, you know, the lack of a relationship between, in some cases, white matter hyperintensity and vascular risk factors doesn't necessarily mean that it's just amyloid or just tau, but there's a lot going on at the level of the neurovascular unit that's vascular inflammatory and interacts in complicated ways with AD pathophysiology in the context of AD. So I think that's sort of what I'm trying to get at a little bit. But thank you for that comment. And Jazz, you'll have the last word on it for sure.
Rin, that was a great comment. I was going to actually make the same sort of idea and expand it out a little bit further, which is that I think the dichotomy between vascular and AD is overwrought. You know, obviously CAA is an example where there's an intersection between amyloid pathology and vessel pathology. I do think that, you know, part of the conceptual underpinning of the Shirzadi paper that you showed up there, which is from my group, is that when we see patients in clinic that have AD and they have white matter hyperintensities that are quite prominent, there's a question: is there an additional disease mechanism at play here? And this isn't just a forgive the pun, an academic question. It means, you know, when we treat people with a medication like lecanemab, for instance, we're worried about side effects. Okay, if somebody has CAA physiology, we don't necessarily want to expose them to this medication. But it's also the case that if somebody has vascular, very strong vascular driver of disease, treating the underlying amyloid process may not do very much for them. You know, it could create a boundary condition for how they might respond. And I think that the point, sort of, for me anyway, with that paper was this idea that if you see somebody who has AD, they have extensive white matter hyperintensities, you don't have to assume that this is a mixed, quote unquote, vascular pathology that has your classic drivers of hyperlipidemia, high blood pressure, all these other things. it could be part of the intrinsic disease process, and you should think about CAA being in play. And in fact, that thought has made its way into the appropriate use criteria for lecanemab as well. But overall, even if you have Wallerian degeneration, you have a change in the blood vessels anyway in that particular location. So I think it's a little bit of a false dichotomy.
Yeah, I think that's an excellent point, and thank you for bringing it back to the patient and the person. So that's the end of our session. There are a number of comments or questions online. Please email me or Antoine, and we'll try to circulate it among the group if you want, if we can respond to those. So thank you all for sticking it out, and we'll see you soon.